This video is an overview of the cell membrane, membrane function, and the cytoplasm of a eukaryotic animal cell. The cell membrane, also known as the plasma membrane, is a membrane in all cells that separates the interior of the cell from its external environment. The membrane is selectively permeable to certain molecules such as ions or organic molecules and is in complete charge of the movement of molecules and substances going in and out of the cell. Its overall purpose is to protect the cell from its surrounding structures and environment. As seen in the picture above, the cell membrane is mostly made up of a lipid bilayer, which also consists of embedded proteins, carbohydrate chains, and transporter proteins. The transporter proteins are what aid in the movement of molecules and substances in and out of the cell. There are also carbohydrate chains attached to the proteins that are embedded in the bilayer, which can be used for cell signaling. Lastly, there is a small compact layer in the middle of the bilayer that consists of phospholipids, which occur with proteins, glycolipids, and sterols to create the space in between. The bilayer is created when the hydrophobic tails made up of fatty acid chains line up against each other, forming the membrane, and form hydrophilic heads on both sides of the membrane facing water. Molecules that contain both the hydrophobic and hydrophilic region are known as amphipathic molecules. The hydrophilic heads of the phospholipids are polar molecules and allow them to interact with polar water molecules. Phospholipids are able to rearrange themselves in the bilayer membrane, which is an important feature in the membrane in order to self-heal itself. Cholesterol makes up about 30% of the cell membranes in animal cells. Like phospholipids, cholesterol molecules are also amphipathic, with a hydrophilic head and a hydrophobic tail. The polar forces allow the hydrophilic regions of the cholesterol molecules to bind with the hydrophilic heads of the phospholipids, while the nonpolar regions of the phospholipids and cholesterol interact by van der Waals forces. The primary function of the plasma membrane is to protect the cell from its surroundings. Due to the components of the membrane, the phospholipid bilayer and the proteins, the membrane is selectively permeable to ions and organic molecules and regulates the movement of substances in and out of cells. Plasma membranes must be very flexible in order to allow certain cells to change shape as they pass through narrow capillaries. The cell can be represented as a castle, and the wall around it is represented as the cell membrane. The castle wall not only provides structure for the building, it also regulates who and what can go in and out of the castle. Also, the plasma membrane plays a role in anchoring the cytoskeleton to provide shape to the cell and attaching to this extracellular matrix as well as other cells to help group cells together to form tissues. It also maintains the cell's potential. This is similar to the castle wall, helping to provide structural support to the castle's overall structure and being used to attach the kingdom to other buildings outside the castle wall. The movement of a substance across the selective permeable membrane can either be passive or active. The most direct form of membrane transport is passive. Passive transport, such as diffusion, occurs without the need of energy from the cell, and active transport requires the use of energy. In passive transport, ions or other substances move from an area of high concentration to an area of low concentration. A physical space in which there is a varying range of concentration of a single substance is said to have a concentration gradient. The cell employs several transport mechanisms that involve biological membranes such as passive osmosis and diffusion, transmembrane protein channels and transporters, endocytosis, and exocytosis. To get more in depth with the process of diffusion, know that as the substances move from an area of high concentration to an area of low concentration, the process continues until the substance is evenly distributed in the system, creating a homeostatic environment. In solutions containing more than one substance, each type of molecule diffuses to its own concentration gradient, independent of the diffusion of other substances. Many factors can affect the rate of diffusion, such as the concentration gradient, the cell's permeability to the substance, the size of the particles that are diffusing, and the temperature of the system. Some materials diffuse readily through the membrane, but others are hindered. Those substances' passage into or out of the cell are made possible by specialized proteins such as channels and transporters. A good example of active transport is the sodium-potassium pump. The sodium-potassium pump is an example of primary active transport. In an animal cell, there is a higher concentration of potassium ions outside of the cell than inside of the cell, and there is a higher concentration of sodium ions inside of the cell than outside of the cell. During primary active transport, the sodium ions are transported out of the cell, and the potassium ions are transported into the cell against their concentration gradient through the use of ATP. ATP is the energy source needed for this pump to function. The sodium-potassium pump is an important feature in nerve cells as it is the primary device used for nerve cells in transmitting nerve impulses to one another in order to send signals throughout the body. Out of all the functions of the plasma membrane, its ability to transmit signals using complex proteins is the most sophisticated. These proteins can be receptors, which work as receivers of extracellular inputs and as activators of intracellular processes, or markers. This allows cells to recognize each other. Membrane receptors provide extracellular attachment sites for cellular influencers, like hormones and growth factors, that will trigger intracellular responses. Some viruses, like HIV, can hijack these receptors to enter the cells, causing infections. Membrane markers allow cells to recognize each other, which is vital for cellular signaling processes that influence tissue and organ formation during early development. The cytoplasm is located all throughout the cell. It surrounds every aspect of the cell and is enclosed by the plasma membrane. It makes up about 70% of the cell's volume. The cytoplasm is a gel-like substance found in both plants and animal cells. The cytoplasm is made up of proteins, carbohydrates, salts, sugars, amino acids, and nucleotides. Its primary function is to hold all the cellular organelles within a cell, as well as important proteins and other molecules. Within the cytoplasm, many chemical reactions which are crucial to life occur. The enzymes in which the cytoplasm metabolize like macromolecules into smaller parts so that it can be easily available for the other cellular organelles like mitochondria. The cytoplasm is also the place where the cell expands and growth of the cell takes place. The cytoskeleton provides the shape to the cell and also facilitates movement. The cytoplasm helps transport as well as protect genetic material in the nucleus. The cytoplasm also helps to transport important proteins and molecules around the cell. It is important to note that the cytosol has no function, but is instead the site in which many processes including signal transduction occur. Organelles are small, membrane-bound organs within the cell. These organelles perform vital processes inside the cell. Some of these organelles include the mitochondria, the endoplasmic reticulum, the Golgi apparatus, vacuoles, and lysosomes. Cytoplasmic inclusions are tiny particles which are suspended in the cytoplasm and range from calcium oxalate crystals to storage granules of materials like starch and glycogen. This presentation has been brought to you by the students of Robert Maxwell from the Georgia State University Biology. Thank you.